बस ठीक है. So, good morning, all. Today, the first lecture is about designing controlled experiments to investigate cognitive and behavioral processes using animal model. So, the first slide basically tells the various factors that can affect the results of an animal experimental studies. So the factors includes the state and health of experimental animals, the test conditions, the level of measurement, data acquisition, level of automation, technical state and calibration of test equipment. Then there are different other factors, trait factors, which involves the strain, subline, genetic background, gender and age of the model organisms. The time of testing, that is day, night, or whatever time of the day that you are testing the doing a particular test. Then the experience of the person that is performing the experiments, and the place where the animals have been kept, and the development conditions like housing conditions, social hierarchy. These all are various factors that affect the results of an animal experimental study. So what's the difference between cognitive versus behavioral processes? So when we do behavioral analysis, we can test two different type of parameters, and in those two parameters as well, we have various other various tests that we can perform for cognitive evaluations as well as for general behavior evaluations. In cognitive evaluations basically involve the mental activities of the brain, such as learning, memory, attention, perception, language, and decision making. They help an organism understand and respond to its environment. So cognitive evaluations basically tell us about the ability of an animal to process in a particular environment, how the brain of an animal thinks. The behavioral processes are the observable actions or responses that result from these mental activities, like exploring, eating, avoiding danger, or social interaction. So the behavioral processes tells us about how the body of the animal acts based on the thinking. So these are the various model organisms that you have already been sensitized about in previous lectures by other dignitaries. So there are different model organisms like C. elegans, Drosophila melanogaster, mouse, rat, and monkeys. And the other image on the side that basically shows that how amyloid beta accumulation and plaque formation leads to cognitive and motor alterations, and how in the preceding slides we will see that how these motor and cognitive alterations can be tested in various neurodegenerative disease animal models. So rats and mice are among the most commonly used animal models in behavioral neuroscience research. The basic reason is that it is easier to evaluate the behavioral changes in rodents rather than other model organisms like C. elegans or Drosophila melanogaster, because you cannot detect various different changes in their general behavior or cognitive evaluations in other model organisms, but it is rather easier in rodents such as rats and mice. From tests of basic locomotor and sensory functions to analysis of more complex behavior related to cognition and emotionality, various types of behavioral tests are being conducted on rodents. These behavioral tests exist for various neuropsychiatric disorders like anxiety, depression, schizophrenia, autism, addiction, ADHD, post-traumatic stress disorder, and neurodegenerative diseases like PD, AD, Huntington, stroke, etc. Okay, so this slide tells us about the behavioral paradigm. Structured experimental setup. So behavioral paradigm is basically the experimental setup that is used to study specific behaviors under controlled conditions, which helps link brain function and observable actions. Its purpose is to model human psychological or neurological disorders and assess learning, memory, anxiety, pain, motivation, and decision making. So these tests help in drug testing, evaluating the effects of a particular drug, how they affect the brain of the animals, and studying disease mechanisms. Like if a drug affects the cognitive ability of an animal, you can ascertain that based on which type of cognitive ability is being compromised, like it is memory, in memory it is working memory or recognition memory, what kind of memory is being affected in that particular disorder. You can ascertain the underlying molecular mechanisms, and the validation of that will be done through the molecular assays that you will be doing on in the ex vivo part after isolating the brain. So okay, so it also, these tests are also helpful in telling that how genetic or environmental factors affect behavior, and the common examples of these behavioral tests that are being widely used in animal experimental studies are open field tests, which are used to evaluate the locomotion and anxiety levels. Then there is elevated plus maze for anxiety behavior, Morris water maze for learning and memory, tail flick and hot plate test for pain response. So we'll start with the locomotor tests. So locomotor tests basically assess the movement, activity levels, and motor coordination in animals. These are often used to evaluate the effect of drugs, neurological disorders, or genetic modifications, and can detect hyperactivity, hypoactivity, or motor deficits. Common examples are open field tests. So in open field tests, there's a large open square arena where you leave the animal to explore the arena, and you check the basic behavior of an animal. Since you know that rodents are, their natural behavior is to explore a particular area when they are left in a new environment. So if there are any alterations from that behavior, they are not exploring. There are several different aspects or parameters that we study in exploring. One of them is rearing. They stand on two paws, and their upper two paws are in the air, and they are exploring all the walls, and they are checking all the things that are in that square arena. They sniff the odor, a particular odor. They explore the arena. So if there is an alteration in that particular behavior of an animal, the animal is not exploring, is showing anxiety like behavior. How will an animal show an anxiety like behavior? If the animal is not exploring, we have left the animal in the arena, but it has gone from its origin point where we have dropped it to some corner, and it sat there for the whole entire testing period, and it is not exploring the arena. So we know that there is some kind of anxiety, or if there is hyperactivity, the animal is exploring more than expected. So how you will compare that? You compare that from the control rats. You, every time you do certain tests, you use control rats as well as diseased rats, then you compare the results of the diseased rats in comparison to the control rats. Your standard baseline will be the behavior of control rats. So the other test, there's rotarod test, which is used for assessing the motor coordination and balance by timing how long an animal stays on a rotating rod. So by time the speed of the rod, we keep on increasing the speed of the rod. We first sensitize the animal of how to walk on the rotating rod by making it walk on a slower speed, and then we gradually keep on increasing the speed to make the animal learn the process of balancing on the rotating rod. So if the animal learns well to maintain the balance in the repeated experiments where we keep on increasing the speeds, so that means that the animal's motor coordination and balance is okay. But in other case, when they are compromised in movement disorders such as Parkinson's, these motor coordination and balance parameters are compromised. So in that case, we will see that how much we try that the animal stays on the rod, but it will keep falling down. You will try again and again, but the animal will keep falling down, and this tells us that the motor coordination is impaired in diseased rats. Okay, then third is wheel running. So these are just the examples, and there are various other tests that are behavioral tests that we'll be discussing in next slides. Wheel running basically tells voluntary activity measured over time to assess motivation and endurance. So the animal is left in a rotating wheel, inside a rotating wheel, and we check that how long the animal is moving and how fast the animal is moving. So the speed, balance and motor coordination are again the things that we test in wheel running experiments. Okay, so this slide shows the setup of an open field test. This is the most basic and common test that is being done for any kind of animal experiments that people do concerning brain, obviously. So this test basically tells us about the normal behavior of an animal, the exploratory behavior. The purpose of the test is to assess spontaneous locomotion and anxiety levels. The procedure is that the rodent is placed in a large enclosed arena and is left to explore the arena to test the exploratory activities of the open field test. And the applications of this test are to check the drug effects, genetic model assessments. So these are the various behavioral parameters that we can study using the open field test. So there's locomotor activity, track plot pattern, center zone entries, time spent in center. So as I already told that more the animal stays in the center of the arena, so it tells us about that the animal, the anxiety is less in that animal. If the animal is exploring, it's staying in the arena, it's going in and out. So that tells us about the absence of anxiety in those animals. So if you see the other columns, you can see that there is a comparison between control rat and PD rat, that how these parameters will be differing in different model rats, disease model rats and control rats, and what is the interpretation for that, what those changes tell us about. So if you see the locomotor activity is high. There is extensive movement across the arena because the animal is exploring the arena properly in control rat, but in comparison you can see that in PD rat there is low restricted movement which is often confined to the corners. So this indicates bradykinesia and reduced exploratory drive. Bradykinesia is basically the slowness of the movement of a rodent. Then we can see the track plot pattern. The track plot pattern is dense, and there is uniform coverage of center and periphery in the control rats, but it is opposite in rotanon or PD rat model. So all these observations are basically based on the test that we have ourselves carried out in our laboratory, and I'll be sharing the videos of the same with you in the towards the end of the presentation. So I'll be showing you the videos of the control as well as the PD model rats and how the behavior changes happen between them, the differences between their behavior parameters. Moving on, this is the overview of rotarod test. So it is used to evaluate rodents' motor skill, learning abilities, and the procedure is that the rodent is placed on a rod that accelerates, and the rotating rod elevated above padded base. So this is just the setup and applications of rotarod test. Applications are relevant for Parkinson's and ataxia research. Then next test is beam walking test. Its purpose is to evaluate rodents' fine motor coordination and balance. In this, the rodent traverses a narrow elevated beam, and there's a narrow beam which is suspended between the platforms, which basically is used to test the motor coordination and balance. So the applications involves the cerebral function studies and neurodegenerative research like Parkinson's disease. Okay, so these are the different behavioral tests for sensory perception. These involve hot plate, one fray, and tail flick. So why sensory perception is important to study? So it tells us about the animal's complete ability to detect external stimuli, process the information through neural pathways, and generate appropriate behavior responses. In pain research, we focus specifically on nociception, which is the neural process of processing noxious and detecting pain stimuli. And why behavioral testing matters? Because behavioral test serves as an essential tool in controlled laboratory environment, and they provide quantifiable, reproducible metrics that bridge molecular mechanism with whole organism responses, enabling researchers to advance fundamental understanding of pain pathways and modulation, screen novel analgesic compounds with clinical potential, and validate therapeutic targets in disease-relevant models. So the first test is hot plate test, thermal nociception. This test basically evaluates an animal's integrated response to sustained thermal stimulation. A rodent is placed on a precisely controlled metal surface maintained at 50 to 55 degrees Celsius, and the researchers measures the latency period until specific pain-related behavior emerge. And what are those behaviors that are observed? Are paw licking or shaking, jumping or escape attempts, vocalization. So vocalization is basically species dependent, and a mandatory cutoff time of typically 30 to 60 seconds prevents tissue injury while maintaining ethical standards. So this is what is kept in mind while doing these tests. So this test primarily engages central pain processing pathways requiring integration across multiple levels. The second test is one fray test. This employs calibrated nylon monofilaments of increasing stiffness. So pain in the form of, you can say, a needle is pricked to the paw of an animal. And the stiffness of that particular needle keeps on increasing with the passing time. We start with a small pressure, but then we keep on increasing in the following tests. So this is used to quantify the mechanical nociceptive thresholds. Each filament delivers a known force measured in grams, so when applied perpendicular to the plantar paw surface until buckling occurs. So the 50% withdrawal threshold is calculated using the up-down method and providing a statistically robust measure of mechanical sensitivity. The clinical relevance of this particular test is in neuropathic pain conditions. Example, diabetic neuropathy, nerve injury. Mechanical allodynia, which is a type of nerve pain, and the inflammatory hyperalgesia following tissue injury. The test's quantitative precision makes it ideal for dose response curves and longitudinal studies tracking sensory changes over time. The third test is tail flick test. So in this, a focused beam of radiant heat is applied to the tail of the animal, and the latency to reflexive tail withdrawal is precisely timed. So this is also a pain evaluation test. So the applications of this involves rapid assessment. Advantages and applications involve: first is rapid assessment. Tests can be completed in seconds with minimal handling stress. Second is reflex specificity. Primarily test spinal circuits, useful for distinguishing central versus peripheral analgesic effects. The third is reproducibility. There's less variability than tests requiring complex behavioral integration. And the safety measures. There's an automatic cutoff, typically 10 to 15 seconds, which prevents tail tissue damage. This test is particularly useful when studying opioid analgesics, as it can differentiate spinal from supraspinal mechanisms of action. Okay, so how to select a particular test for our research that we will be doing. So there are several parameters to consider. So selecting the appropriate assay, it depends on the research question, what you want to study, the neural pathways of interest and the pain modality being studied. Different tests have different applications and different uses. Like hot plate test is particularly useful if you want to check the do the opioid screening or central pain processing. One Fred test for neuropathic pain and allodynia studies. The tail flick test is important if you want to screen analgesics and spinal mechanisms. Okay, this is just a further extension of the applications and impact. The first one involves the fundamental pain neuroscience. Elucidating the molecular and cellular mechanisms underlying acute and chronic pain states. So these assays enable the researchers to map nociceptive pathways from peripheral transduction through spinal integration to cortical perception. The second is analgesic drug discovery, so screening novel therapeutics and their effect in reducing the pain, or maybe if they're increasing the pain, to induce the pain, what kind of effect the novel drugs are showing. The third is disease modeling studies, characterizing sensory abnormalities in models of neuropathic, inflammatory, cancer-related and post-surgical pain. Longitudinal testing tracks disease progression and therapeutic responses over time. The last is neural circuit analysis, dissecting the specific contributions of peripheral nerves, dorsal root ganglia, spinal cord circuits, and supraspinal centers. Combined with optogenetics and chemogenetics, these tests reveal causal circuit behavior relationships. Then first we saw about the motor coordination test, normal anxiety exploratory test. Then we saw about the pain related tests. Now these are the learning and memory function tests. The first one we'll be discussing is Morris water maze. So in this, a rat or a rodent is basically left in an open water tank to explore the arena. And there's a hidden platform in the open water tank which we want the rodent to identify. And the first time we place the rat in that arena, the rat takes longer to find the hidden platform, because why it finds the hidden platform is because it has to, otherwise it has to keep swimming for that duration. Therefore it finds the platform to go into the resting phase. So the first time when you place the rat in this water tank, the rat takes longer to find the place where the hidden platform is. But in the preceding experiments, when you again put the same rat in the same conditions, you will see that the rat has developed a memory that where that platform is, and it will straight away swim to that particular place to reach the hidden platform. So this tells us about the that the memory impairment in this particular rat is not there, and this is a control rat. So if you want to study the learning and memory, this test plays an important function, and this can be useful in the study of neurological disorders like with cognitive components such as schizophrenia and Alzheimer's disease. So I've already told you the procedure for this. So this image basically shows us the phases that I have told. In the first phase, you can see that the rat takes, we have dropped the rat here. This is the origin point. The rat takes longer to find out the hidden platform, which might be here or here, right? In the second test, we have dropped the rat here, and it straight away goes to that particular place where the hidden platform is. The third one shows impaired memory function in a rat. So the rat is exploring the arena, but it is not able to find the hidden platform. Okay. So the next test for testing the spatial learning and working memory is radial arm maze. So in this, there's a central platform, and there are 8 arms around it, which is used to assess the working memory. As in Morris water maze, cues are placed around the room. There are different cues placed in different arms of this maze to distinguish, for the animal to distinguish between different locations. That the animal will know that there's a particular reward placed in one of the arms when we placed for the first time. The animal will explore completely, and it will know that in these particular arms there is reward placed for it, and the next time you keep the animal in that, it will develop a memory, and it will straight away go to that point where the reward has been placed for the animal. So okay, the third one is Y maze test. Like radial arm maze, there were 8 different arms. In this one there are three arms. Okay, so this test is basically used to evaluate the spontaneous alteration behavior in rats. And in this, the rat, for the first time, the rat is placed in an origin arm, as you can see here, and the animal is allowed to explore the arena. All 3 arms are open for exploration. In the second phase, when you will keep the same rat here, you will block one arm, and you will allow the animal to explore the 2 open arms. So since the normal behavior of animals is exploratory behavior, they explore the novel areas where they are placed. So when you in the third phase, you will again open this arm, and you will see the frequency with which the rat is visiting this novel arm, because for the rat, this arm is the area that the rat has not explored. Right, so the frequency of entering the novel arm will be more in comparison to the other arms if the memory is not impaired. So this is Y maze test. Next is novel object recognition. This is again a memory evaluation test. So in this, what we do is, in the first phase, we place the rat in an open arena. The arena is similar to that of open field test. There's a square open arena where we place the rat, and there are 2 similar objects placed in the arena. The animal will explore both the objects in this phase, and in the next phase, what we do is we change one of the objects with a novel object. So we see the ability of the rat, or the time the rat will spend with the novel object, because of its normal exploratory behavior. The rodents possess. So if the rat's memory is not impaired, it will spend more time with the novel object rather than the similar object. If it has gained a memory that I have already explored this one, it will not spend more time with that, but with the other one. So this is another memory test. These are the various parameters that will be evaluated using the novel object recognition test. The first one is the exploration duration, which is high. in control rats because it actively explores both the objects. And in PD animals, it will be low, which tells us that there's reduced motivation and cognitive impairment in those rats. The object preference in control rats will be that the rat will be spending more time with novel object rather than the similar or the familiar object. But in PD rats, there will be brief approach to familiar object only. These results are again based on the results that the experiments that we have done in our lab, and I'll show you the videos for it. So these are all the observations for control rats and PD model rats. So there was a brief approach to the familiar object in case of PD rats, but however in the control rats they explored both the objects but spent more time with the novel object. So there are various interpretations based on it, which tells us that in PD rats we saw impaired recognition memory, bradykinesia, reduced exploratory drive, and cognitive decline, motor impairment, etc. Then there are tests for anxiety and depression like behaviors. The first one is forced swim test. So in this this test basically is helpful in identifying new classes of antidepressants and anxiolytic drugs, as well as discovering genetic factors linked to mood disorders. The test uses immobility as a measure of behavioral despair. So the procedure involves the animal is placed in a cylinder filled with lukewarm water. A normal animal will initially display swimming and escape behavior, but will gradually spend increased time immobile, that is floating. However, a diseased rat will show more anxiety-like behavior as it will keep trying to escape the cylinder by showing climbing like behavior. It will keep climbing and try to escape, and there will be more anxiety in the diseased rats as compared to the control rats. So okay, so that was all about the different kind of behavior test, and now we'll be talking about a software. This is Animaze behavioral tracking software, which is basically used to test the behavior of animals. This okay, so most of the people do the behavior test manually. So there are various variety of alterations that a manual test can give. It could be based on the person that is performing the test. Okay, so there could be various different changes than the normal behavior of the animal that you can see in a manual test. However, the Animaze is an automated software that can give us the actual behavior of the animals that they are displaying, and we can test at a particular time. If we are performing one test, we can get a number of different parameters. For example, if you are doing open field test, you place the rat in the open arena for around 3 minutes or 5 minutes. In the normal manual test, you will only see if how many times the animal is rearing, how much time it is immobile, that is it has not moved, how much time it is taking to explore. Okay, so but in when you use these behavior tracking softwares, you can get the precise number in seconds, time in seconds, that how much time the animal took to explore the arena, the number of in and outs, like it went to the center and then came out, how much time it spent in the corners of the arena. You can basically make different grids, specify different zones to test that in one particular zone how much time the animal spent. In different zones, you can compare the experimental parameters in between different zones in one single test only. So its purpose is to automatically record and analyze animal movement, interactions and behavioral metrics. It is widely used in rodent studies to test different behavioral assays like open field, Y-maze, and novel object, etc. The core features of animes involves flexible video tracking, works with the live camera feed or pre-recorded videos. So you can use both. You can either record the videos live, or you can feed pre-recorded videos as well. Supports many camera types. There are simultaneous multi-camera option as well available as well. You can track the whole body or head or tail alone, or you can do the partial tracking as well as whole body tracking. So as I already told, you can define different zones in the apparatus, and you can test speed, immobility periods, path efficiency, head orientation, rotations, etc. You can do manual scoring, or take the notes from the automated scoring that you will the data you will receive after the test that you are doing. These are the various applications. You can use anime software for all different kinds of behavior tests like open field tests, Y-maze, NOR, anxiety, lighten, dark box, elevated plus maze, water maze, specialized assays, etc. Okay, so this is a little video of anime software, how to use this software. I'm sorry. So this part is basically defining the walls of your apparatus. You are defining the walls in between where you want to do the analysis. Okay, so using anime software is also beneficial for publication purpose, because when you do manual behavior testing, you have recorded the data yourself. You don't have any proof that you did this particular test or how the results were. Have you manipulated the data or not? But in this case, you have recorded validation for your results. Once if the reviewer asks you that can you give any proof that you did this particular test and you received these results only, and there's no manipulation of the data, you can always show them the videos. There are track plots and heat maps also that you can get from this particular software, which you can show to the reviewers or anyone that asks for the validation of it, of the behavioral data that you get. So this is again a video that I mentioned earlier. This is the OFT open field test video of the control animal. The first one is the actual recorded video of the animal during the test, and this is the track plot. You can see the behavior of control rat, that it is exploring all the corners of the arena. It will explore properly. This is the rearing pattern, that when it stands on its two paws. Okay, so the next one is the same experiment for a PD animal model. So you can see the difference between the exploratory behavior of control and animal model. So you can see there's slowness of movement, bradykinesia, anxiety-like behavior. The control rat kept on exploring for the entire experimentation duration, but you can see that this rat, from the origin point, it just went to one of the corners and it will keep sitting there for the entire experiment duration. So, okay, so this one is heat map and track plot representation of control and rotanon. The first, the same one for the from the video I just showed you. The other one are the different treatments that I gave in my own study, and checked the effect of those treatments in improving the animal behavior. So, okay, so these two are the different doses of different tests, different treatments I gave, and these are the standard drug. Okay, so this one is NOR video for control animal. The round object here is the novel object, and the rectangular bottle is the similar object. Okay, so this one is for the PD animal model. You saw in the control rat that it explored both the objects as well as the whole arena, but spent more time with the novel object. And this is the diseased animal. So this animal went to the similar or the known object earlier, and now it will start displaying anxiety like behavior by staying in one corner of the arena, and it will not explore the novel object. So that's all about the behavioral test and anime software. If you have any questions, you can ask about the software or different behavior tests.
How frequent do you do this training tests? Like during training, how frequent do you retake? And how many times in a day? If a more is always very continuously.
That depends on your experiment. So in our research question, we created like we did habituation first. We habituated them to the arena. Then after 1 hour, we did the test. So it is important to habituate the animals to the arena when we will be doing the experiments, but the frequency that how long we want to keep them training them for that.
Each period.
Sorry, yes, we have to train each and every day separately.
And for everyone?
Same for everyone. The experimental setup and procedure that remains same for every kind of treatment. It may be control or rotenone or maybe a treatment, different treatment drugs that you are testing.
This latest parameter should be not be same for every treatment.
We try to do the test for all of the animals in the same particular way because once you go to another day, your apparatus, because you have to set the anime software each time, you have to give all the parameters or the specifications to the anime software. You have to draw the entire apparatus in the software to basically define the area in which you will be doing the experiment. So that changes a little bit every year. So if you want to compare the results of control to your treatment group or to the animal model group, you have to perform the test of the same way.
Is there any difference in the different tests used to study the same aspect? Like there is a UAT plus space test, there is LIDAR box specific, they are all coincide with the same. Like why
They
they
basically validate the result of one test. Because in particular test there would be various variations that you can see because the animals want to perform in a similar way in each test. So in one test you might see a particular behavior of the rat that you might not see in another test. That depends on the various different factors that get worked out. If you are performing the test, you perform one test early morning when they are active or during the night. If they are nocturnal, they will be active during night. If you do during the afternoon or during any time of the day, you will see that they are not performing that well. So you will get different results at different time of day and various other parameters they and factors they also play a role in the behavior of the rats. So basically different kind of tests are available to validate the result of one test to the other. And the ultimate validation is done on the basis of molecular resistance that you perform after the dissection.
if you're physically inducing some computer environment, for example, so there are two groups, there is control and there is sham. baseline? Which one should be considered
That
the
depends
baseline?
on your, again, on your model, experimental model, animal model. If you, in your model, for example, this one, the work that I just told you about, this PD model, I induced the PD with rotenol, right? I did not do any kind of surgery to induce my model. Only if I did any kind of surgery, I surgically injected the rotenol in particular brain region or any other thing to induce Parkinson's disease. In that case, I had to use sham. Why? Because in sham, I'll be doing the same surgical method, but I'll only be giving any kind of buffer or something.
Now there will be a control group too, no? So which one should be considered the baseline trial disease? The control one or
I
the
think
sham?
that you should compare the results with sham, because that will give your you the good results in terms that if your surgery is responsible for any kind of behavioral change, if surgery is doing any behavioral change, that will be visible in both sham as well as your other surgical groups as well. So in that case, I think sham should be the baseline.
And while doing behavioral experiments, have you encountered any challenges like variations in the results?
Yes, in behavior tests there are a lot of variations. I myself performed these tests, I think, n number of times. At first, I was not getting any results because, as I already told you, that different factors play a role. I used to do my test at any time of day. I started doing that in the middle of the day, during the afternoon. The animals.
But again, they are nocturnal animals. If you do the animals during the night, you are doing it for all the group. So even if the performance is reduced, the error, they will be uniform in everyone. Like 4 groups if you are taking, you are doing it at night. For all the 4 groups you are doing it at night. So now ultimately the result should be comparative. We draw the significance on the basis of comparison between the groups. So do you think when there is really a factor, the timing of the group?
Okay. Timing is one factor, but I think to answer your question better, the number of animals in particular group, that also matters, because they are after rodents. They will behave differently every time when you place them in a particular arena. But if you have a larger animal, the number of animals, you are using 8 to 10 animals in a particular group. So there's a probability that more than half of the rats will give similar results, but 2 to 3 rats will always give variable results. So that is why we use higher number of rats and we apply stats for that purpose to minimize the error in the data that we are getting.
If you are doing 10 animals, how many outliers do you usually expect or accept?
Outliers, there may be 3 to 4 animals in a particular test that may perform differently than all the other ones. So for that purpose, you might have to repeat the whole entire group another time. Again, if you are not getting any animals, you may have to change the set of animals. You have to get fresh new animals and you have to perform the test again.
If you are performing the test and it's the same animals, then what is the validity of it? Because it is based on remembrance. So they have already explored the whole apparatus. So next time they will have some memory of the apparatus and the result will be very different.
Yes, and one more adding to the question. When we have all the established models working on MPDP, then these are all day-based. For example, if we take 1 day for, we will take 1 day for to carry out the whole Y-way spontaneous alteration. Then noble arm, if the group is more, it might go till 2 days. Morris water will take 5 days and the probe trial will take 6 days. But if we are supposed to repeat whole Morris water again, then we are increasing the number of days of the experiment. So is it good, ma'am? Like now we need the result, we might have to publish if we extend it. Sacrifice days will also get extended, biochemicals and all.
That you have to plan your experiment, I think, because animal tests, they take time. You have to give time to behavior test, and it is not significant to ethically significant to do more than 3 to 4 behavior tests on one particular set of animals, because they will not perform. If you do keep on doing 10 tests on one particular set of animals, they will not perform. They will only give validated good results, the results that you want, in 2 to 3 tests. So choose the best tests that are really important for your study and perform those tests. If you want to do another set of tests, you may have to do the entire animal experimental protocol again and test the other parameters that you want to test. But again, the control animals will be different and the model animals will be different, and you will compare the model animals to that set of control animals, to the new test that you have done.
Ma'am, in your this period while you were doing your project period, did it ever happen that even after performing twice the behavior, you like had failure and you had to replace the entire set of animals?
Right, yes.
It happened, right?
Many number of times, and I performed behavior animal test. Maybe 2 to 3 years of my study was based on animal test, and I performed again and again the test, because I was not getting, there were different changes that I did to my study. I tested, like I was not getting the results, so I had to change different parameters, test different parameters, that what is the problem that was there in my test. I had to identify the problem, and I had to re-perform the tests to get the ideal results that I should get in comparison to the literature that I saw.
But when you do it alone, ma'am,
Most of the tests alone, but in the initial phase, I had to get some help,
because
because
ma'am, the in my case the problem which I've encountered is that while clicking and all, you are setting the animal, then you some time goes while you go and just click in the software, you lose some time in that period.
yes,
So what do
you have to be fast enough,
Yeah.
and you have to, you know, uh, learn to do the test efficiently, because at the end of the day, PhD is your own independent work, and you have to learn to do your test independently. In the initial days, even I took some help, but no one helps for more than one particular period, you know that.
Ma'am, one more question. After taking all the measurements in a control group, then if in case if we have to do stereotaxical measurements, then after inducing a drug directly to the brain, how much? What are the indications that by then we have to measure all the parameters?
You are saying that after how much time of the surgical
Procedure.
procedure you have to do the behavior tests?
Yeah.
That is based on the literature. I think you can get better results that in how many days the disease is being induced after the surgical procedure from literature, and you will take that as a baseline. You will perform the behavioral experiments on the last day, that is the period of the what do you say study timeline, right? So that for that you have to do the literature survey and check that what is the minimum number of days in which the disease will be induced, and you can get the symptoms in
Do
behavior
we have
tests.
to check these parameters every day?
No,
Or
no, that every day you can check every day as well, but that also depends on your study, because in my study was for a duration of 35 days, and I checked thrice during that duration. Before the start of the experiments, to check that all the animals are performing like control, right? In through the midway, and then at the end. I did it thrice, and that also depends upon your study design. So you will go through literature review and see how how people are working in that particular area, and how you want to move forward with your study. That all depends upon the research questions, your disease model, and the study design. No, I did not use Vonfre, but one of my fellow lab mate is using Vonfre, and she is using in UIPS, Punjab University, pharmacy department.
So that's a like a sophisticated situation where we use.
Right, right. It's not available everywhere, so that's why she's also working in UIPS for behavioral analysis. Any other questions? Thank you.
So I'll just not spend too much time on introducing the disease, probably since you are all a cohort with similar interest, maybe you know it. We always been saying that, that neurodegenerative disorders involve protein aggregation disorders. This is also a protein aggregation disorder. Alzheimer's disease is a protein aggregation disorder. Parkinson's, Huntington, or disorders of protein aggregations. There are other pathologies also described for the first time 1817, and further this disease was characterized by Charcot in 1800s, and this was a description of this disease, or you can say publication about the disease, an essay on shaking palsy. Motor functions are impaired, that we all know, and so motor dysfunction was described something like cerebral palsy. These are cardinal features, hallmark symptoms: resting tremors, bradykinesia, muscle rigidity. Few more, but I was just saying incidence all across the globe. Primary pathology involves depleted or low levels of or loss of dopaminergic neurons. When I say dopaminergic neuron, you can see the picture on the right. If there are less number of cells, they will make less amount of dopamine. Imbalance of dopamine versus acetylcholine is what is creating this muscular or cerebral motor palsy that we were describing. And it happens primarily in this area called putamen or substantia nigra. Putamen caudate nucleus is the area where dopaminergic neurons are there. Maybe all of you are neuroscience, we all say different regions of the brain are different tissues actually, because they behave very different, chemistry is different. Interestingly, there are non-motor symptoms that you will see in the early phase, and then you will get motor symptoms little late in the time course of the disease development. And we all know probably this disease is a mitochondrial disease. Mitochondrial complex 1 is important. A lot of studies support this transgenic overexpression of PGC. So if we have a PGC1 knockout mouse, they are more vulnerable to the toxicity of MPTP, which is a toxin that depletes the cells in the brain. And if you overexpress this PGC1 alpha controls the mitochondrial biogenesis. So number of mitochondria depend on this. Anything that increases or activity of this PGC1 alpha or amount of this PGC1 alpha will be beneficial. It also talks about resveratrol, which also acts on PGC1 alpha and increases mitochondria. So other supporting evidence. In interest of time, I am not getting into this. This is talking about existing therapy. What are existing therapy? You are supplementing dopamine from outside, fine. But we know there is a problem with that, because your enzymes to metabolize will also be going up. Either you will have to decrease the dose, or another strategy is I can give an inhibitor of monoamine oxidases or other enzymes that are involved in degradation as an adjuvant, so that because peripheral system will also metabolize dopamine. So the kind of therapies it says is L-DOPA is one that is given. It talks about dopamine agonist, which will mimic dopamine, monoamine oxidase B inhibitors, then I am involved in degradation of dopamine. Carboxy O-methyl transferase inhibitors can also be useful. So all these can be possible therapies, and it is talking about the sites or biogenesis or dopaminergic system on the left side. I am not getting into this. So all these existing therapies have adverse effects, because these are all chemical agents. If you look at it, the dopamine agonist, some examples here, then COMT inhibitors, monoamine oxidase inhibitor, sertraline, resgalene, few more, L-DOPA we all know, and there are a few more of these types that we can talk of. All right. Adverse effects. So this is just a literature to build the story that there are problems with existing therapies. So we stumbled on this molecule. Compare the molecule on the left, compare the molecule on the right. So it's one amino group is replaced by hydroxyl group. We said let's compare maybe this. At least it would do something to the scheme of things where dopamine is involved. Mainly present in olive oil, virgin olive oil, potent antioxidant activity, and it's been used as cardiovascular protector. So I said this already, structural similarity to some extent, at least the basic backbone was similar. Maybe I can put that down. All right. So key questions were, does hydroxytyrosol have any anti-Parkinsonian targets? Can it rescue dopaminergic neurons? Can it be neuroprotective? Can it affect mitochondrial functions? So the first part was in silico. I will not get into too much details, but you can see we looked at binding of hydroxytyrosol, rasgalene, which is, I said, is monoamine oxidase B inhibitor, other molecules, even dopamine, it does show interactions with monoamine oxidase B. We also did it with monoamine A also, and compare that and look at something very interesting. This is hydroxytyrosol, this is rasgalene. Sites where it binds are almost kind of identical. So that means pocket that is using is also similar. So whenever we talk about this binding, we also do molecular dynamics, which says the complex that is formed is stable, and this just was validating that part. All right. We also look at blood-brain barrier permeability, and then we also look at Lipinski's rule of 5, which says molecular weight less than 500. Other properties, hydrogen donor, acceptor properties, all fits into the criteria that we are interested. It is also blood-brain barrier permeable also, which also supports this hypothesis that maybe there won't be any issues of crossing blood-brain barrier. So I'll just say that maybe whatever I said is already explained. I'll skip this also. So now we wanted to validate whether it will do something in vitro or in a real situation. First part was in silico. So I would say why we started doing in silico. I never did in silico. This was a shift we had during COVID time because labs were closed. So we all decided let's do something based on in silico. So what we did was we wanted to look at whether it will work in an actual scenario. So we took platelets which is rich in monoamine oxidase B and then screened this enzyme activity. We also looked at whether it will have any effect on synuclein polymerization also using thioflavin T fibrillar assays. So this is the concept we are saying, platelet isolated, look at monoamine oxidase B inhibitor reactions. And maybe I'll just take a call, something, give me a second. Hello. So this is the kinetics you can see, compare hydroxytyrosol versus rasgalene. So IC50 is 4.82 nanometers. For hydroxytyrosol it was in micromolar concentration, higher, but this is a phytochemical, that's a synthetic drug, there has to be differences, but it does inhibit monoamine oxidase. That means it can be inhibitor of monoamine oxidase B. Even synuclein aggregation, you can see hydroxytyrosol, this is synuclein aggregates. When you treat it with hydroxytyrosol ratio 1:4, you can see aggregation is, this is TEM scan, and the top is the fluorescence signals to pick up where it says aggregation is prevented when the ratio of hydroxytyrosol to synuclein was 1:4. So two sites where we could show that it does inhibit in vitro. So we wanted to go take the studies to in vivo system. Two models we used, C. elegans and MPTP mice model. Why C. elegans? I will just talk about it briefly. Certain number of neurons, 308 precisely, and you can map all these neurons. Dopaminergic neurons 8, all can be seen individually because optical clarity of the worm is also there. And you can use these mutants. N2 is a wild type mutant. NL5901 is a mutant where you have the fluorescent protein, yeast fluorescent protein being expressed in the muscles. I'm sorry. And BY555 is a mutant that has GFP in dopamine containing neuronal soma and processes. So I can see dopaminergic neurons in this C. elegans. CW152, which doesn't have complex one. So one model which is a mutant of PD, this model is where GFP is being expressed in the muscle. There is another model where you are expressing a protein in the muscles, and this is a wild type mutant. So we wanted to see whether there is any toxicity of hydroxytyrosol. No toxicity at all at higher concentrations up to something like 500 micromolar. And this is synuclein aggregation assay. This is done in the strain called NL5901, where we said this fluorescent protein is being expressed in muscles, and this is synuclein protein. What you can see is this is control, higher fluorescence. I will just talk about where the significant difference is. You can see green fluorescent is gone down, indicating that it prevents aggregation in the muscles, which is an evidence in favor of hydroxytyrosol being useful. So likewise, yeah, that's just highlighting the bar. So other thing that we want to tell you is monoamine oxidase is also an enzyme required for activated converting MPTP, which is a toxin, to its active form, that is MPP. So this enzyme is present in astrocytes primarily. So when you inject MPTP into rats, this has to get converted to MPP, which is neurotoxin actually, and it will kill those neurons. All right, this is just a background. So you will, this strain which has bright GFP fluorescence coming from dopamine containing neurons, if I treat them with MPP, it should kill those neurons. Am I right? Because I am only seeing bright fluorescence from dopaminergic neuron. I treat it with MPP. What you should see is, all right, this is control, strong fluorescence from the dopaminergic neuron goes down, recovery with hydroxytyrosol, although not 100%, but significant recovery after hydroxytyrosol. So it's rescuing dopaminergic neurons from MPP induced toxicity. So another test that is done is called neuronal repulsion time. Repulsion time is linked with content of dopamine. Repulsion time, so you give this chemical, the worms would repel because this is an irritant for the worms. So with this compound nonenal, once you give, what you see is this is the controls. This is the repulsion time. I give MPP, there will be loss of dopaminergic neurons, repulsion time has increased, and with hydroxytyrosol this time has gone down. That means dopamine containing neurons, functional assay of dopamine in the C. elegans. This is done using NL5901 strain and BY555 strains. So I can also look at mitochondrial viability with the strain that was GAS1 mutant. So GAS1 mutant is where the mutant did not have complex 1. So you can see activity is low, hydroxytyrosol mitochondrial fluorescence is higher in these cells because mitochondria has been seen using mitotracker deep red staining in this cells. So I am just not getting into all the data for this, maybe we can skip some, and then we also wanted to look at what happens to the expression of these complex or mitochondrial genes that will be involved in PD also. So we looked at PDR1, LAGR1, PINK1, ERK1, SRK1 in this NL5901 strain. You can see the activity has gone up in one which are beneficial, gone down in the case which are not good. So maybe just summary of what I will say. The mRNA expression shows hydroxytyrosol reduced synuclein toxicity in the strain of C. elegans by upregulating expression of PDR1, LAGR1 and downregulate expression of GRK1. Rest of the things I already discussed with you. We also wanted to go to in vivo model, the rat model, because sometimes it is helpful. Use different doses of MPTP, pre-treatment, post-treatment with a hydroxytyrosol. The dose of MPTP we used was 20 milligrams per kilogram body weight, 4 injections in a single day. That's what is the accepted dose for creating this model. Something important, we have to use here C57 black 6s. We have a data where we compare two strains of mice and we say this C57 black 6 is the only relevant model. So one of the behavior tests for seeing whether they develop PD is catalepsy, which they are asked to made to walk on a bar, and MPTP treated animals day 16. You can see the descent latency is increased significantly, indicating motor function is impaired. Narrow beam walk test, maybe interest of time, I will say you challenge the animal to walk on a narrow beam, which wider at one end gets narrow at the other end. So it's a challenge for them to walk if their motor functions are impaired, and the one end is baited, and you can see there is time to cross the beam is increased. Say day 16, hydroxytyrosol has been beneficial in them also, and probably we did more of behavior test, and one of them is pole test, and I'll not get into data because I was told we can do a quicker session on this. Maybe if anybody wants, we can discuss that. Rotarod also, probably many of you have done this test. One test we do in our lab for motor functions is called footprint analysis. Normal animals would have hind and forelimbs overlap. MPTP, you can see this overlap is problematic. Post pre-treatment beneficial, they come back to the same gait is restored in the animals. All right. Neurotransmitter levels. I'll be interested in few things that I'd like to talk to you. Dopamine depletion, restoration, hydroxytyrosol not detectable in these tissues which have not been given. You can see hydroxytyrosol in the brains once you treat that. You can estimate hydroxytyrosol in the brain also, and significant increase in these animals. We can also look at monoamine oxidase activity. It is suppressed in the treated animals, indicating it also does inhibit the activity also. So anyway, so you can also look at dopaminergic neurons by tyrosine hydroxylase staining, and immunohistochemistry reveals there is a reduction in the brain, restoration. This is reduced tyrosine hydroxylase restoration in the brains after hydroxytyrosol treatment. So I will not get into repetitive data. This is quantification of the tyrosine hydroxylase dopamine transporters, indicating the same kind of findings that it does restore dopaminergic neurons. Nissl stain. Then we also looked at mitochondrial functions. They are restored after giving hydroxytyrosol. This is done using oxygraph where you look at oxygen consumption and then you can look at different complexes of mitochondria. This is the data from there. You can also look at NRF2 protein, which is an antioxidant response gene, and we can see if I just compare MPTP and post hydroxytyrosol, you can see significant difference at the level of Western blot also. Mitochondria swell, MPTP toxicity, they are restored. Electron microscopy also says something similar. All right, mitochondrial biogenesis genes, PGC1 alpha, suppressed significantly following MPTP exposure. Recovery significant happens after other genes like NRF1, which is also involved in mitochondrial biogenesis. TFM also shows the same trends. So almost similar kind of things. This is again Western blot for some of these genes in the same order. We can also look at mitochondrial dynamics. I'm not getting into that. So we can say it favors. So if I have to sum up what all we could say based on our study, we could say hydroxytyrosol prevents synuclein aggregation. It prevents degradation of dopamine. Antioxidant also functions, mitochondrial functions, and all this would contribute towards having beneficial effects in PD. So we are not saying this is a treatment. We are saying still this is a lead molecule. You can improvise this molecule, or you can use this molecule as an adjunct to existing therapies, because it has pleiotropic properties, antioxidant, mitochondrial biogenesis, few other properties. So if we have to say treatment, we still have to work on this molecule more, and this is the work primarily being done by Anjana, who was a PhD student and now works for a company in Bangalore. And maybe since I have slides, I forgot to talk about this. I also represent one of the entities on the campus called Northern Region SNT Cluster. You might have seen this logo also in our certificates. It's called PI Rahi, Punjab University, IIT Ropar, Regional Accelerator for Holistic Innovation. Why I wanted to talk about this is, we all the time should not be interested in publication, maybe think of something which is innovative, which could translate into something real. So we say regional SNT cluster, we have a mandate of this region of India, J&K, Himachal, Punjab, Haryana and Chandigarh also. And it is a section 8 company, that means we are out of the bureaucracy of Punjab University. Punjab University and IIT Ropar are 50-50 stakeholders into the company. These are our thematic areas. Anybody having an interest can come back to us. Agriculture, food processing, pharma, healthcare, medical devices, any indigenous technology, development and optimization, sustainable mobility, green energy, waste management and waste from wealth. So we do support startups, we accelerate startups, anything which is innovative, which can be a regional solution provider, which can help this region. So we have a lot of programs that we launch. We do capacity building. So anything under science and technology umbrella is part of it. We are the 7th cluster in the country. All others are city-based clusters. The recent addition cluster is an interesting addition, and I probably think that maybe you've not heard that name. Maybe explore that. AMTZ, Andhra Pradesh Medical Technology Zone, which is a huge space, 270 acres. You can say Sector 14 campus of Punjab University could be similar space, where they only talk about medical devices coming from that area. So look at the kind of technologies they come up. Delhi has a cluster called Drive, Pune has a cluster called PKC, Bangalore has a cluster, Bhubaneswar has a cluster. So we are the 7th cluster, and this idea of cluster also came from Chandigarh. We had an entity which Dr. Gurjaspreet represents called CRIC, but CRIC, we are now an official, you can say, and legal entity which got recognition in the form of cluster and recognized by principal scientific advisor to Government of India. That means our reporting is to the Prime Minister's office. And thank you for your time and attention. I did discuss short because of what the thought was. Anybody with a question, I would like to answer that. So I just wanted to give a flavor of what we do. We keep on evolving in terms of our interest, and we do multiple different areas of research. You also, I think, to the mathematics. Anybody else have a question? Yes.
What is the mechanism of action of this hydroxy So you said it is helping in looking also you said that it has a structure similar to that of dopamine. So is it competing with dopamine to interact with the different receptors?
That's what we think. That it is. So it has multiple targets. It itself is antioxidant, which I did not highlight in my study. So we started with looking at if dopamine is what is the target. But if you look at literature, it says it also targets mitochondrial biogenesis. And even I would say, if you look at more recent literature, people may have found more targets of this molecule. I said, if you look at initially when I said it is a cardioprotective molecule, some literature suggested it is beneficial for mitochondria also. It has good effects on biogenesis of mitochondria. All kinds of not known. So pleiotropic properties are something that I will say. But one target is definitely monoamine oxidase B, but there are other targets also, which could be secondary or primary, that we cannot say. What we used monoamine oxidase B from human. So in crystallized structure is required. I think you probably, maybe Dr. Dindima did talk about that. You need a crystallized structure that makes life easier. If you don't have, then it is a bigger challenge. You will have to then think of some other approaches to find it. Or you can predict the structure. Probably there are alpha-fold site probably that can predict the structures of the molecule. Probably can use it. I'm not having that much of expertise in silico work, but we collaborate with somebody from BHU who has high-end computing facility. Because for doing molecular dynamics, you need high-end computing facility. If you don't have it, then probably there is a challenge in having good results. So they have a bunch of supercomputers there in BHU that they use. We also have a supercomputer on the campus, but probably we are not using them for in-silico.
Sir, in silico, like we have done previously, like we are only limited to know the admit profile of emitter if we know the gate component, nothing more. Nothing more in silico other than admit profile.
You can know a lot of things. Even the enzymes that will metabolize it, the n number of properties you can examine from. Not from exact precisely admit, but there are other tools along with that which will give you more information on pharmacodynamics of the molecule also. So more can be done. Nothing. But Nidima is the best person probably in the campus who can tell you all those other tools that can be used. Maybe we can also connect you with person with whom we collaborate. Maybe tell you all other tools that can be used. I also kept my in silico work very precise because the meaning I wanted to convey is that I wanted to give a flavor of it rather than doing deeper into each part of the segment. I didn't show all the analysis that we did for considering. There are more tools we can use. So one name in a thesis can be in silico also. So it tells you, gives you some leads. Anybody else with a question? All right. Thank you. Any more questions?
It's okay to start. It should work. Okay, so the last lecture will be just the research presentation about one project, one of our last projects, which includes rare neurodegenerative disease, mitochondrial function, and drug development or drug target research, and a lot of imaging. So I'm going to specific details, even if they are maybe not so interesting. I think they are still valuable to understand what is required to be, how to say, sure about your conclusions and how difficult sometimes it is to do something. Some of the slides you have seen already before in previous presentations with pictures. So one of the diseases we are studying is this Wolfram syndrome. It is a genetic disorder, and it is characterized by diabetes, and in addition to diabetes also optic atrophy, deafness, and brain atrophy. It is considered to be also neurodegenerative and neurodevelopmental disease. Neurodegenerative because the brain stem is degenerating, at least, and also maybe other areas as well. Diabetes in these patients could be managed more or less with current therapies, but the problem is that the neurological symptoms can't be managed. There is no drugs, and usually these are the main problems for patients. They will first get diabetes, this could be managed, but then the optic atrophy and deafness will start. They anymore can't see properly, can't hear properly. So, and this usually happens already when they are 10 years ago, sometimes maybe in 20s, but mostly when they are relatively young, and life expectancy is maybe to 30s, sometimes to 40s, and it's quite, how to say it simply, quite bad disease as other such kind of genetic neurodegenerative diseases. So there is no real treatment, and the reason why there is no treatment, as in case of other diseases, is that even if we know the original protein, we don't know exactly its function. If we don't know the function of that protein, then it's quite difficult to find some kind of targets also. It's not monogenic disease. There is two genes involved. One is the wolframin, which is giving the name to this disease. It is, you can see the multipass protein in endoplasmic reticulum membrane. It has big portion inside of the ER in lumen, and it has also big domain in cytosol. And it's big, 100 kilodaltons or so. And the mutations are in many different places. So you see these red dots, this is the protein, red dots are, how to say, mutations. So you have mutations in many places. And it's not that one mutation dominates, you have a lot of different mutations in patients also. This is making the gene therapy quite complicated because you have your therapy against all these mutations. So at the moment it is not realistic to develop such kind of gene therapy because of the cost and because of the regulations. Then another protein is CIS2 that encodes this ZDGSH iron sulfur domain containing protein 2. This is simple protein, single base protein, but it is a more interesting protein because something is known about its function. So it is binding the ferrum sulfur clusters, you remember from the mitochondrial function lecture. It interacts with BCL2, means it could control apoptosis, autophagy, and it is somehow interacting with SERCA, reducing SERCA maybe, and it is necessary for SERCA activity. Most of the disease cases are related to mutations in this wolframin 1 and relatively few less to mutations to this CIS2. In symptoms there is slight difference. The CIS2 deficiency has few more symptoms. And CIS2 is also interesting because it is such kind of anti-aging protein. It has been shown that at old age its expression is decreasing, and if it is activated in mice, then it is, how to say, making the life of old mice better. It's also, if you overexpress it, you activate it, it helps in case of Alzheimer, it helps in case of cardiac problems in mice. So it's such kind of anti-aging protein, and actually people are studying it because of that, not because of its involvement in Wolfram syndrome. So two very different proteins, but they are doing at this, in cell more or less the same things. If you don't have this wolframin 1, then you have ER stress, you have ER calcium disturbances. It's difficult to sell which is coming first. Then you have a mitochondrial dysfunction, then you have a mitophagy and huge effect on cellular bioenergetics. And CIS2 is doing absolutely same things. ER stress, ER calcium problems, mitochondrial dysfunction, mitophagy, and problems in cellular bioenergetics. And they are interacting quite same proteins. So we started to study this actually already 15 years ago, and then we showed, then people were thinking that it is ER protein and it's ER stress disease or ER calcium disease. But the symptoms still are mitochondrial disease symptoms what we have. So then we showed that actually it's the mitochondrial dysfunction that's lead to this disease, that the key factor which is progressing that disease is in mitochondria. So together with us after several papers came out showing that it's related to ER mitochondrial calcium fluxes and contacts. So but we were still interested because it was not so clear why this ER calcium problems or ER calcium flux to mitochondria, why this happens. And if this happens, then why and how much it really affects the mitochondria. Because a lot of people, even after these papers, they were thinking that mitochondria were secondary players, just they became affected, but in disease progression they are not so relevant. And then, since we are from pharmacology department, we also try to find targets for treatment. So the first question was that why you have less calcium in ER and going to mitochondria. So it is quite tricky to study. So the model we used the primary cortical neurons from 1-day-old rat pups. Why you use specifically these neurons? The reason is that they were, in our case, they were, how to say, disease-relevant cells. Because if we are looking for the drug targets in case of neurodegenerative neurological disease, then it makes sense to study neurons. And since axons are the most affected part in these cases usually, we focused also to axons only. So we didn't study the cell bodies or somas, but we studied this end of the axons, which is one of the most important places if you have neurodegeneration or neurodevelopmental issues. Then we used the specific plasmids encoding the siRNAs to suppress this wolframin 1 or cis2 in our model in these cells, and then we measured everything related to calcium. ER calcium, cytosolic calcium, or oxoplasmic actually, mitochondrial calcium, ER to mitochondria, calcium flux, and contact sites. So, in every different place which was relevant in our context. So you saw this picture or similar picture. So here we have axonal membrane is red, and these green lines, they are ER tubules. So calcium is in these tubules. We used ratiometric dye to have a good normalization, and as you see from this, just this control graph, if we treated the cells with glutamate, this released ER calcium, and we have significantly less calcium in ER, so the dye was working well. Only thing is that to find this specific dye, this 210 means here the calcium, how to say, affinity. We had to spend more or less a year to test all different dyes, which will, not dyes, sorry, it's genetically encode sensor, to find the sensor which is perfect in our case. And we did it the same for cytosolic or oxoplasmic calcium. We measured the oxoplasmic calcium. So these measurements alone, they have no value. The value comes only if you quantify these measurements. So here 1 dot means 1 axon. So we picked up the average axons and we measured the calcium level ratiometrically at the end of axon in cytosol or in endoplasmic reticulum. 1 dot, 1 axon. So what you clearly see here is that in groups where we do not have a wolframin 1 or we do not have cis2, we have much less calcium in the endoplasmic reticulum. And in the same groups we have much more calcium in the oxoplasm. These differences were not so visible in cell soma. In soma everything is well balanced, buffered, controlled, and the differences were relatively small or missing even. So what this graph says? This says that we have such kind of situation that we have less calcium in ER, more in cytosol, but nothing more. So this is what is the limitation if you have just this graph. It's just description. It's not showing the reason. So to find the reason why you have this difference, you need to manipulate with system. So What we did, we first expressed the pump, Cerca pump, which pumps calcium from cytosol to ER. What we saw that if we will overexpress this pump, then we have more calcium in ER and less in cytosol. So it's not perfect. You see, this is the wolframine Sirna, and this is the same together with Cerca. There is such kind of 15+% effect. The same is in the case of this tool Sirna. So this means that these two things are related, really, that it's not two independent things, that we have less in ER and more in cytosol. No, it's because of, it's not maybe because, but it is at least related to the calcium pumping. So one reason why we have not enough, we may not have enough calcium in the endoplasmic reticulum could be also that it is leaking out. But okay, I forgot this figure. We made one more experiment. We actually overexpressed phospholamban, and this phospholamban is suppressor of Cerca. And this alone made situation very similar to what we observed in disease models. So, but okay, so another thing why we don't have enough calcium in reticulum is that it may leak out from reticulum through the release channels. So first we tested this ryanodine receptor calcium channel in ER. So we took the siRNA to suppress this channel, and it was again protective. We suppressed the leak, and calcium normalized in reticulum in cytosol. So until that everything was logical. We don't have enough calcium in endoplasmic reticulum, it's not pumped in and it leaks out. Then we went and we tried another channel. Is it also responsible for leak? So we suppressed the IP3 receptor that is in ER membrane, but it was not giving any protection. So there was no improvement. So either it's not involved in leak or it plays some other roles. So the next question is that, okay, we had more or less the reasons, not enough calcium, but we wanted to see is that the real reason why we don't have enough mitochondrial calcium. So we used similar sensors to measure mitochondrial calcium, ratiometric sensor, and measured it in individual mitochondria. What we saw, it's okay. We saw that we have less calcium in mitochondria, which was expected. But then it was not showing exactly how much this is again related to disease and so. So we made such kind of more complicated control experiment. So we took yes, you can hear now me also. So we suppressed the wolframine 1. As a result, so we had less calcium in mitochondria. Then we reintroduced wild type wolframine 1, which is siRNA insensitive, and we restored the mitochondrial calcium levels. But when we used the mutant wolframine 1 plasmids, which expressed disease mutations, then in some cases we got no rescue, and in some cases we got perfect rescue, similar to control. So we had 4 mutations. What is important here, that these 2 mutations which were not working, they were from the patients who have severe early, relatively strong, early starting Wolfram syndrome. And these 2 were from patients where the symptoms were mild. So we showed that it's indeed disease severity related effect what we have. Okay, so then the next question was that, yes, it was still description. We have less calcium in ER, less calcium in mitochondria, but we don't have a link between these two things. It's not causatively linked. So what we did, we improved calcium level in endoplasmic reticulum. We overexpress TERCA to have more calcium, or we suppress the ryanodine receptor to have a more calcium. And we measured the mitochondrial calcium. Mitochondrial calcium was going back to control level. So this is saying that there is causative relationship. Problems in ER, even corrected, they will lead to mitochondrial improvement. Means that ER is the primary reason. But then we had very strange results with IP3 receptor before, and we decided to check what happens if we increase the IP3 receptor activity. And what was slightly unexpected, that when we increase the IP3 receptor activity, mitochondrial calcium also increased. If we use the dead IP3 receptor, it became even worse. So when we wondered why we have different results, why there is protection, then we were thinking that it is because the IP3 receptor is giving the calcium directly to mitochondria, but ryanodine receptor most likely gives it not to contact site but to cytosolic part. So they are giving calcium to different compartments. So what we concluded then? There is less calcium in ER but more in cytosol or oxoplasm. It can be rescued if we will promote calcium uptake. It could be rescued if we suppress the leak through ryanodine receptor. But strange results with IP3, and we were thinking that it's because of the mitochondria ER contacts. So you saw this figure earlier that we have a sensor. This was originally designed and published by a Japanese research group, which is very good to measure the mitochondria ER contacts. That's the nanolook is the most sensitive, the luminescence. Most sensitive luminescence producer, luciferase assay. So we used that assay, and what we saw, yes, we have less contacts between mitochondria and ER. For some reasons, less. So it's good, but it's again description. Less contacts, but is it relevant? Maybe it is, maybe it's not. So we tried to design control experiments. So one control experiment, what we designed, is that we tried to increase the number of contact sites. We express the GRP75, which is one of the proteins keeping the contact sites together. We overexpressed that. We got more contact sites in control. We got more contact sites in Wolfram syndrome model, as expected. And then we measured mitochondrial calcium. What is good? So you see Wolfram syndrome, mitochondrial calcium less. We increased the number of contact sites. Calcium was coming back to mitochondria. So what is the conclusion is that we don't have enough calcium or enough contact sites for calcium movement from ER to mitochondria. One or both. So then what we also did, we measured the direct calcium flux from the ER to mitochondria. So induced ER calcium release by specific compound and measured how much goes to mitochondria. What we saw is that significantly less calcium is going from ER to mitochondria. Control here, disease, wolframin Sirna here. 3-4 times. And also we saw that with cytosolic calcium. So we really don't have enough calcium in the ER. So we can't release much. If you can't release, or there is nothing to release, then mitochondria will get nothing. So we established what happened from here to here. So we had a problem, not enough mitochondrial calcium. So it's nice, but so what? Is it relevant at all? Is it affecting, how much it is affecting the mitochondrial function? So we measured everything we could. Membrane potential, mitophagy, mitodensity, ATP, NADH. What we saw, the membrane potential, so there are different dyes. This is now dye, not sensors. DMRM, GC1, GC10. So GC10 has its advances because it's ratiometric. If you have a low potential, then it's green. If you have a good potential, then it's red, and the ratio shows how good potential you have. But it has also limitations. It is red, starts to aggregate. If you wait too long, concentrations are too wrong, then everything aggregates to red. So you should catch the point where everything is still measuring something. And another thing people don't know so well, that it's toxic. In neurons at least, if you use it in concentration that is suggested, then your mitochondria will become round shaped. They are dead, almost dead. So to do this experiment, you should use the lower than suggested concentrations, and you should always check what happened with your mitochondria. Are they still happy? Otherwise, you are having in all groups semi-dead cells. So we did that, and what we saw that we have slightly less membrane potential, or less GC10 red to green signal in disease neurons. Not much. If we treat with uncoupler, then it goes to 0. So some people may say it's not much, but for the membrane potential, it's quite a lot. You can't expect, if you have disease and the carrier of this disease is still living, so it's not so strong disease. You can't expect changes from here to here, because then it's not possible to live anymore. Membrane potential changes in these disease models are always very small. If you use pharmacology, then they are strong, but in the models you can't expect this big change. We have less potential. Then we have mitophagy. So we measured using this gamma probe, about what I was telling yesterday. We measured that we have much more mitochondria inside of lysosomes if we have a disease models. We counted it. One dot is one cell, means how many dots here we had in one cell, and then this red circle is showing that number. But very big increase in disease models, almost twice more mitochondria inside of mitophagy. Means mitochondria were unhappy. What happens if you have too high mitophagy? It's mainly it's if you have a mitophagy defect, then it's bad because you have accumulation of bad mitochondria. But if your mitophagy is for some reason very strong, then one thing that happens is that you have less mitochondria in cell. It removes so actively the mitochondrial population that you have less mitochondria left. So mitochondrial density is going down in both of these disease cases. It's not only reflecting the mitophagy, but it's mostly, I believe, in our model it reflects mitophagy. And when mitophagy is difficult to measure, you need good microscopes, sensors, then the mitochondrial density or quantity is easy to measure. Simple microscope image, we use the sensors, but you can use the fluorescent dyes. You will stain, you will count, or you will measure somehow how much you have. Easy to get numbers out. Maybe time consuming, but cheap. Then when we discovered that we don't have enough enough mitochondria, then what is next to measure? No mitochondria, no energy. So we use this Perceval probe to measure ATP at the end of axons. So it's again ratiometric. The red is the axonal membrane. Axonal last segment of axon, and this is the presynaptical terminal, or maybe it's just a growth going at the moment, which is soon forming the presynaptic terminal. It's a relatively big, good to measure place in axon. So we measured it here. And what we saw there is that we don't have a significant decline in ATP. So this was most, one of the strongest effects we saw. No ATP at the end of axons. When we measured the ATP in soma, the effects were small. Means that the neuron body could be happy, but everything happens at the end of axons and in synapses. It's really necessary to measure what happens there, not what happens in body. The classical ATP measurement is the biochemical assay from all cells. In this case, the biochemical assay shows you absolutely nothing, because problem is in this place. And compared with all signal, this is very small part of signal. So biochemistry will not help, you need imaging. Then what happens in cells when you don't have enough ATP? Because of the mitochondrial suppression. Cell will die, try to do more ATP through the glycolysis. Then you have a lactate accumulation, and you have NADH accumulation in cytosol. So it's another name for that is Warburg effect, which happens in cancers, or people call it reductive stress also. So we use different sensors to measure, do we have more NADH per NAD? And again, we got, in both cases, results showing that there is more NADH in disease groups than in control. So these probes are more tricky. The ATP probe is working always, but with paradox and sonar we have sometimes problems in neurons. This paradox, Perceval, GIAN, calcium sensors. They are all available from GIAN. If you need them, you can find them from that GIAN. The sonar, at least a few years ago, was not there. This I got from the Chinese research group who made it. But then it sometimes takes time to get these probes, because from GIAN it's 1 week you get bacteria, you will grow them up a few more days. So it's 7 days as approximately from order to your plasmid. If you're asking it from another group, then there is paperwork, there are contracts, and it could take months to get it. So the repository, even you pay something there, is always faster. So we made conclusion that because of lower calcium, we have lower membrane potential, lower ATP production, less calcium in axons, who activate glycolysis. We have redox stress because of that. So what remained open here is that how exactly these mitochondrial calcium problems were leading to lower membrane potential. So it was the question, and it was, how to say, we had two independent mechanisms or pathways, but they were not exactly together at that moment. So we were asked to explain that. So what happens, you remember you had pyruvate dehydrogenase in lecture before. So this pyruvate dehydrogenase and actually also other these mitochondrial dehydrogenases, they are calcium sensitive. If you have a calcium, then pyruvate dehydrogenase becomes more active. If you have less calcium, no calcium, then it is passive. So, and this is related to phosphorylation of the pyruvate dehydrogenase. So what we did, we tried to check that if we manipulate with pyruvate dehydrogenase, then what really happens. So how to make pyruvate dehydrogenase more active? You can inhibit it, of course, but in our case, we wanted to activate it. And you don't have pharmacological tools or good tools. You can overexpress it, of course, but it's not, it shows that pyruvate dehydrogenase will be relevant, but it's not showing that it is inactive or not enough active. So we overexpress the pyruvate dehydrogenase phosphatase, which removes the phosphor from pyruvate dehydrogenase and makes it more active. So what we got partial rescue. We made the pyruvate dehydrogenase more active using this approach, and this is, you see, the ATP level control, disease, and we got it to somewhere in the middle. When we measured NADH levels, again, control disease, and with phosphatase somewhere in the middle. So it's not strong evidence, not strong enough evidence, but it shows that it plays some role at least. So if it would have been negative, no rescue, then this means that this mechanism has nothing to do with our problem. Then we also wanted to check, is these problems, NADH, etc., ATP, are they related directly to mitochondrial calcium? So we overexpress the meroprotein, which is known earlier by others, by us, to increase the mitochondrial calcium. We increase the mitochondrial calcium, and you see indeed, mitochondrial calcium in control, disease, and we overexpress mero in disease, we got it back. And same with ATP, control, disease, mero, ATP is coming back if we increase the mitochondrial calcium. So we got direct link, mitocalcium, ATP, NADH. Then we did the same at the level of ER to mitochondrial calcium flux. So we overexpressed the IP3 receptor. And the idea was that if we overexpress it, then more calcium is going from ER to mitochondria. So controlled disease, IP3 receptor, half is coming back, dead receptor, nothing is coming back. So it shows that the ER to mitochondrial calcium flux was partially at least the reason for the ATP loss and so on. And we did the same with Cerca and a few more experiments, and we made conclusion that ER calcium handling defect and ER to mitochondrial calcium flux are the reason for everything that is here. Then again, if you are speaking with medical doctors or clinicians studying that same disease from clinical, perspective, they will say, who cares? So you have in your dish less ATP somewhere, but it's quite far to say that it's relevant. So since it's neurodevelopmental disease, in addition to neurodegenerative, we studied how well the axons are growing in these neurons. So control neurons, axons are growing. If we will knock down the wolframin or cis2, axons are not growing so well. Axons are growing significantly less. So this means that we made a control experiments later also to improve it. And what this means that it's not just some minute effect in our ATP signal, but it has really relevance at the end for the neuronal, not function, but neuronal morphology, which is always affecting its function. Then one of the sub-aims we had there is that we were always studying two proteins, and they always did the same. Both disease-inducing wolframin cis2, when we knocked them down, they always, you remember from the figures, gave us absolutely similar phenotype. How this is, why is the reason? There was one old paper showing that they don't interact. You can't immunoprecipitate them together. But at that time, the antibodies were bad and so on. So we were actually asked to find out how they could do the similar thing, how they are related. So we started from colocalization experiments. You saw this yesterday, that wolframin is in the ER and in axonal terminal, and very similarly cis2 is in the same place. We made colocalization experiments, yes, they were in the same place. Then we also made immunoprecipitations. We started with immunoprecipitations with the artificial constructs. So we overexpressed tagged cis2 and tagged wolframin. We got the interaction, but 2 points. The journal nowadays, they don't accept you overexpress interactions. It's good to have them, to show them, but you should always have also proof that there is endogenous interaction. So we tried to do it. I think we tried 10 different antibodies because mostly they were bad. And then, at least at the end, what we got is that when we immunoprecipitated the cyst2, 15 kilodaltons, then the wolfram in 90 kilodaltons came also with it. It's a tricky to do. You need a good controls. We had here very simple EGG control, but the problem is if you study if you IP cytosolic proteins, then it's easy. But if you IP the proteins which are in cell in the same membrane, in mitomembrane, in EM membrane, then always when you purify your preparation, you will get the membrane fragments at some point which will bring both proteins together to your IP. So you should have a good controls to be sure that this is not that problem. What do you think, is it a proof to show that they interact if we will IP them? It's not, because in this IP mix we have also a lot of other proteins. We IP'd ryanodine receptor, we IP'd SERCA. In one of the paper published after us, they IP'd IP3 receptor. And in all cases, the wolframin is coming together with ryanodine, SERCA, IP3, and cyst is coming. So what we can conclude from this experiment is that maybe they are not directly interacting, but they are in the same complex, and they are in the same complex with main calcium handling proteins, SERCA, ryanodine receptor, IP3. If you want to have data that they really interact with each other, then actually only way is to purify both proteins and to show it. But how to purify exactly wolframin, and so it's not so easy business. We are not strong in biochemistry. We're not doing that, but journal was happy. And okay, they interact with each other indirectly at least, but they interact also functionally. So that if we have here ATP measurement, control cells, wolframin knockdown, less ATP. If we will increase CIS2 expression, we overexpress it, we will get rescue the wolfram deficiency. Or if we have CIS2 deficiency, less ATP, we have a rescue here again. So they are functionally fully interacting at the level of ATP and also at the level of the axonal growth. So axons are not growing, wolfram sirna, they start to grow if we will include CIS2, we will overexpress CIS2. So it's difficult to explain what this means, this interaction. No, really, it's not giving really good conclusions. It just allows to speculate that they are controlling together other proteins. They are controlling maybe SERCA, they are controlling maybe ryanodine receptor, maybe or maybe not, but clearly IP3 receptor. So they are controllers, maybe independently. And what they could sense, speculation again, wolframin could sense ER stress, CIS2 could sense reductive stress because it has these ferrum sulfur complexes. They are maybe sensing this stress, and accordingly they will modulate calcium uptake. This is speculation, of course. And then at the end we had pharmacology. We took all these proteins which we had at the beginning till mitochondrial calcium, and we started to look for the compounds available which are affecting this. So it's SERCA activator, CDN 1163 is available. Then there is azomolan, which is inhibiting guanidine receptor. So here we have more calcium uptake. If we inhibit guanidine receptor, we have less leak. Azomolan is very similar to tantrolan. Tantrolan is the drug used in rare cases to inhibit guanidine receptor. But azomolan is strong, it inhibits it, maybe too strongly. Then there is new experimental drug, Rucal S107, that is not inhibiting really fully guanidine receptor, but leak more specifically. It's also in studies, in some kind of, I think it's in clinical studies, in case it was or is, against the post. I don't remember what exactly it was, but there was some neurological disease. Then we took sigma-1 receptor agonist that increases the IP3 receptor-induced calcium flux. And then we used the diabetes drug liraglutide that has very various effects. I mean, it's clear what it is doing, but it's not so clear what it is doing in neurons, why it affects neurons, how much receptors neurons have. And one speculation is that it is increasing the calcium fluxes somehow. And then we had an inhibitor of sodium calcium exchanger, which pumps or mediates the calcium flux out from mitochondria. So if we inhibit this, then the calcium will remain in mitochondria. So we had altogether 1, 2, 3, 4, 5, 6 compounds. We didn't study this because one group was publishing Science Translational Medicine paper showing that it's protective and also in vivo protective. We continued only with other compounds. So not going to details, we made huge amount of experiments, 2 years for one PhD student. What we got, we measured how these compounds affect, they were all affecting calcium, but how they affect the mitochondrial parameters, mitophagy, ATP, and axonal growth. What we found that they mostly are protective. They mostly give rescue. You see that mostly statistics is quite okay. Only in few cases we don't have statistics. And even in these cases, it is maybe because the drugs were sometimes toxic in some models. We didn't spend much time to find optimal concentration, so we used what was most practical according to literature. Sometimes we had toxicity problems. But in general, all these compounds affecting differently calcium, they were affecting positively the mitochondrial and neuronal health parameters. So the main conclusion then was that for neurons with this disease, the calcium problems are main thing, and that theoretically at least these calcium problems can be corrected. Is it really possible with these compounds, with the exception of liraglutide, which is actually used nowadays off-label in case of Wolfram syndrome? All other compounds are commercially old compounds, commercially available, and you really can't develop them further as a drug. So, but it's still valuable because you have a target. You know that if you have another SERC activator or another guanidine receptor blocker, then maybe it could help. So it's not the drug research, it's a more target research. And okay, it's a rare rare syndrome. It's 1 per 500,000. How many patients? 30,000 patients in the world. But why it's relevant, as you remember, this is the mitochondrial calcium contact site disease, and most of our neurodegenerative diseases and also several rare neurodegenerative diseases, they have this MUM component, which is maybe relevant. So that if we have these targets, then we also can test or speculate that if we modulate these, they could in some cases be used in case of other neurodegenerative diseases. So if you want to know more about methods, then it is now published. More or less a year ago it was published. So you can search my name, Wolfram syndrome, you will get it. And editor was quite happy because we had a lot of different components in that paper. First, the paper was not descriptive, but it was showing the cause, that this leads to that, which is good. Another reason was that we had 95% imaging, and it was quantified, how to say, it is not images, but everything was quantified. And third, that we did something which is, how to say, relevant also, target research. So the editor was happy and included us to this editor highlights, so the last year best papers in the journal. Okay, and we are a relatively small group. We were big 5 years ago, now it's 4 people, 4 PhD level researchers, 4 students, and this specific work was done together with Gert Boltnik, who is from Catholic University at Leuven, from Belgium, who is specialist in calcium. Then with Fingwen Chai, who is from Taipei and who discovered the function of CIS2, and with my postdoc supervisor, who is now retired, with Professor Wexler, who knows everything about the mitochondrial respiratory chain. And also our animal house, head of our animal house, who helped us. We had no in vivo models here, but we used sometimes the neurons coming from wolframin deficient rat to do control experiments. So that is all from me. If you have whatever questions about this presentation, please ask. So it says quite specific, I think, but so it's not relevant to remember what one protein or another protein was doing. It's just good to remember that descriptive science is not any more popular. You should have cause, you should prove that this is leading to that. Okay.
So this was definitely done for a disease condition, which is extreme case scenario, I would say. But what we can we use this further in order to, in general, enhance the mitochondrial health?
Mm-hmm. So it's difficult to say, because if we just overexpressed SERCA, or we did all these treatments with our compounds, then we always had a control group where compound or treatment was alone. And it was, in most of the cases, with the exception of when we had toxicity, there was no difference compared with control. So that if mitochondria were okay, or reticulum was okay, then these compounds had no effect alone. So you can't improve happy neurons, happy mitochondria with these compounds. So one question is that people frequently ask is that this is all cell culture. So where is the animal study? But you can't really do the animal study, which includes so many different components. You can't study the mechanism in animal study because animal studies are very complicated. If you want to test something specific, if you want to test is this compound protective, then you will do animal study. You will use different models and zebrafish, mice. You will show treatment is working, etc. But if you have so many compounds, so big pathway, then there is no hope to do in vivo. Of course, it's the best of the best is to do this, to find that, one drug is really good, and then to do with this animal study. But then it takes not, how long it took, 7 years, but then it takes 2 times 7 years. And this is too much because the grant cycle is 5 years, so we can't study, we can't postpone the publication so long. So this is a problem that is nowadays. When I was a young postdoc, then we made circulation research paper, which is more than Impact 10, with 3 months. And we had all together there, not sure, 10 or 15 image panels. 3 months, 15 image panels, no really big work. Okay, discovery was nice, but nowadays you want to have a good paper. It's 100 plus figure panels. It's not 3 months. If you are lucky, then it's 3 years. Usually it's more than 5. And PhD students, or even worse, postdocs, they have only 4-year, 2-year periods. They really don't have time for big papers. So therefore we are lucky we have 4 staff researchers who can spend a lot of time to do these things. And PhD students, the first author was PhD student, but for her it took also almost 10 years. But since it was not problem for her because she was she's medical doctor and was working part-time in clinics, and it was more or less hobby for her. So no more. Yeah, please. I will come closer, then I will hear you better.
So you mentioned that ATP depletion causes reductive.
Yeah.
Particular threshold of ATP in cell at which point reductive stress runs to oxidative stress.
Okay. So this is a very good question because we can only speculate. The question was how much inhibition in ATP production we need to induce reductive stress or oxidative stress. So, and I can't tell you actually, because it's also a little bit speculation. What happens if you don't have enough ATP? Then glucolysis activates, you have more NADH, but how much you should inhibit mitochondria or ATP to get that, and how strong reductive stress you will get, nobody knows. Even this is speculation that this suppression of ATP is leading to NADH increase, because we really don't have direct evidence to say that they are strongly related. So it's unfortunately it's a good question, but it's bad answer. Okay, then thank you. easier to find the PhD positions because people don't expect from PhD perfect experience, perfect match. It could be slightly different field as well. They just expect that the PhD student is clever and knows theory behind it. It's more complicated, and PhD is depending 3, 4, 5 years. For postdocs it's more complicated because from where you could find that there is vacancy. So usually there are these databases everywhere, okay, Nature Job and others, you know probably them very well. And if you have advert there, then they usually expect quite specific experience. If you are autophagy lab, then you expect somebody with autophagy knowledge. Or if you are not autophagy lab but plan to go to autophagy, then you, or just this IPSIS, you want to introduce them, then you are surely you need somebody who is doing that, having experience. So they are expecting somebody with specific experience very frequently because it's only for a few years, 1 to 3 years, and there is no time for learning because they need it immediately. And there is also no freedom because there is specific project behind that advert you should do. If you want more freedom, then there are these Marie Curie fellowships, there are EMBO fellowships. I don't know the rules exactly, but the competition nowadays is very high because they were thinking this year the competition, the European Commission had 60% more applications, and the reason is maybe that now it's easier to write this non-scientific part, which is because of ChatGPT. Because unfortunately these Marie Curie applications, they are 10 pages long, but unfortunately, how to say, half or something is such kind of not science. But the evaluators are anyway evaluating everything, and this not science should be also written very well. And from here, for you it might be difficult to understand what exactly they expect. If you have ChatGPT, you will get already some help. But yeah, so if you plan this write in future these applications, then it's not just science. All these other, sometimes strange things should be also very well explained. But if you have this, it's good salary and it is freedom also. And in that case, you can choose your lab and your project better. You just contacted someone saying that you want to ask with them the Marie Curie, and then I think in most of cases they agree with that. And maybe there are some other fellowship agencies in specific countries there are. So this is another option to find the lab, to find the fellowship, and to apply. You can also write directly to the labs you like, but this is, I don't know the percentage, but its efficiency is maybe only 1% because usually they don't have vacancies. And they get so big amount of such kind of letters that they are maybe not reading them even. Especially if the field is different. If the field is very similar, then it's another thing, because if somebody with very knowledge which is exactly what I need and I have a vacancy, then I will reply, of course, but usually I don't have money and vacancies, so it's tough. Okay, so no more questions to me. So what we should do now?
We'll have a feedback from the students,
Okay.
and then we'll
With this, we conclude the 5-day GIAN course on molecular insights of neurodegenerative pathways and its drug development strategies. During this course, several different topics were covered around neurobiology, along with the tutorials making the students more aware about the in silico drug designing, proteomics, and much more. I'd like to thank the teaching faculty of the course, Professor Ellen Kasik from the University of Tartu, Estonia, Professor Rajat Sandhir, the coordinator of this event, Dr. Tanzeer Kaur, the coordinator of this event, Dr. Nilima Dhingra, Associate Professor UIPSPU, Dr. Ashutosh Rai, Department of Biochemistry. The lectures were truly engaging and helped all the participants in getting more knowledge and a fresh perspective to neurobiology. I'm sure that we all would be able to incorporate these in our research projects as well, be it mitochondrial studies, flow cytometry, animal behavior analysis, in silico drug designing, and proteomics, to name the least. We all are leaving with a bag full of new research domains to explore. So on the behalf of all the participants, I'd sincerely like to thank all the teaching faculty. Along with this, I'd also like to thank the local coordinator of GIAN, Dr. Professor Gurjaspreet Singh, for organizing this event and arranging everything. Thank you, sir. And now I'd like to call upon volunteers for the feedback.
Hello everyone, myself Tanu Pandey. I am presently doing my PhD from Central University of Punjab, Bhatinda. Now I extend my heartfelt thanks to the organizers and to the speakers for sharing their valuable expertise and giving us a valuable knowledge in this field. Indeed, this was a very good workshop. We got a deeper insight in molecular mechanism, and we got to know new therapeutic approaches. Thank you for this, providing us this platform. Now we are going back with a fresh memory. We are more energetic now. We are more confident now. And we expect that we will have such workshop in future as well. Thank you so much, everybody. Thank you, organizing team. Thank you, speakers, for providing us this platform. Thank you so much.
Hello everyone, I am Anvi Pandey and I am from Naiper Mohali, and I'm currently working under Professor Shyam Sundar Sharma, who is also working in neurodegeneration as well. So when I registered for this program, I had expected that I would get some knowledge regarding the pathways, but what I had not expected was how much in silico work I would get to know. For example, the various processes like autophagy and the various workshops that we did. For example, the the various in silico process that we did, that was a very new thing for me, because in pharmacology we were always exposed to like real time animal work. Also the behavioral lectures that we got today and many other lectures, they were kind of eye opening for me. Also the proteomics, that was quite fascinating and a very novel thing for me. So I'd like to thank all the various respected faculties, Professor Allen sir, Tanzeer ma'am, Gurjaspreet sir, Rajat Sandhir sir, and all of us who imbibed us with such valuable knowledge that will get to cherish us all through our life. Thank you so much.
Hello everyone, I am Pankaj from Institute of Nanoscience and Technology, Mohali, and I am working under Dr. Jeevan Jyoti Panda. I got this opportunity to attend this workshop, and I am very thankful to Punjab University and the organizers for giving us this opportunity. And I would say that this serves a very good purpose of bridging the knowledge gap which exists in subject specific domain, and I would like to we would like to have these kind of workshops in more subject specific domains. Thank you. And I would like to thank Tanjeer ma'am, and Sandhir sir, and Alan sir. Thank you.
Hello everyone, I'm Chaitanya. I'm from BITS Hyderabad. We work on mostly ER mitochondrial organelle crosstalk. Once we came to know about this GIAN course, which is which is also talking about mitochondria and all. So I thought of applying it. Am I here? So for this, I sincerely thank Dr. Tanjir Kaur ma'am for giving me this opportunity to attend this GIAN course, and it's very wonderful learning experience. It's truly we enjoyed all the lectures so far, and also sincerely thank Professor Alan Kasik for giving his insightful ideas about. For me, it's very important for my study because of mitochondria study, related studies, and it's very, and for also thank all the lecturers, all the professors for the interactive sessions. Thank you.
So thank you to all the volunteers for your feedback. Now I'd like to request Dr. Tanzeer Kaur, Professor Gurjaspreet Singh and Professor Ellen Kasik to please felicitate Dr. Ashutosh Rai. Thank you, sir. Now I'd like to request all the participants to come forward and receive their certificates. Kunja Chaitanya, Vikram, Tanu Pandey, Tokir Ahmed, Sandeep, Sangeeta Kumari, Anupriya Rana, Bhavna, Abhishek Sarkar, Anvi Pandey, Barsha Bhardwaj, Nishant Arya, Rajat Chaudhary, Riya Amit Chabra, Ishita Bhardwaj, Tanisha Rohela, Preeti, Kirti Arora, Priyanka Bhardwaj, Shivani, okay, so Ramya Malhotra, she is also not there. Sonia Rani, Divya Rai, Payal, Pankaj, Shalini, Navpreet Kaur, Rimaljot Singh, Payal, Shivani Sharma, Abhishek, I think you're okay, it's not there. Okay, it's not. Sorry. Vanisha Sen, now I'd like to request the coordinators and Professor Ellen Kasik to please facilitate Dr. Nilima Dhingra. उसके बाद Now I'd like to call upon the local coordinator of GIAN, Professor Gurjaspreet Singh, to conclude this event.
A very good afternoon to this distinguished gathering of researchers and students. As we come to close this enriching 5-day workshop on molecular insights into neurodegeneration pathways and drug development strategies, it is indeed a fitting moment to reflect on the fruitful discussion, knowledge exchange, and collaborations that have unfolded over the past few days. First and foremost, I would like to extend my heartfelt gratitude to our esteemed Vice Chancellor, Professor Renu Vig of Punjab University, Chandigarh, and Dr. Anil Kumar, National Coordinator of GIAN, IIT Hyderabad, for their invaluable support for conducting this program. I am especially thankful to Professor Alan Kasich for gracing us with his presence and for sharing his profound insights and expertise with all the participants. My sincere appreciation also goes to our course coordinator, Dr. Tanjeer Kaur from Department of Biophysics and Professor Rajat Sandeep from Department of Biochemistry for their unwavering commitment, guidance and tireless effort in making this workshop a resounding success. I would also like to acknowledge the entire organizing team, especially Mithun, Vineet, Manraj, for their meticulous planning and dedication and hard work, which ensured the smooth conduct of every session. Finally, I wish to convey my sincere appreciation to all the participants for your keen interest, active involvement and valuable contribution during this workshop. I truly hope that you will carry forward the knowledge, idea and inspiration gained here into your future endeavors. Thank you all once again and I wish you continued success in your respective fields. Thank you.
So thank you everyone. This concludes our event. I'd like you to join us for a cup of tea. Thank you.